Hello, everyone. So this will be my last biology review on this channel, likely. At the very least, the last exam review. As I'm a senior and I'm graduating in like a week. So, with that being said, let's get started. Translation. Translation is the act of turning an mRNA into a protein. Just plain and simple, right? And we could derive this principle from the central dogma of biology. Right? So the central dogma of biology states that for any for all of the biological sciences to be true, there needs to be one specific thing that needs to be true where we could build off everything else. And that is, we go from DNA to RNA to proteins. That's it. DNA to RNA to proteins. Right? So what essentially that means is that when we go from DNA to RNA, that is transcription. And when we go from RNA to proteins, that is translation. Right? So, pretty straightforward so far, right? We've learned about a little bit of transcription, right? So, take for example, right? I write something on the board, right? If we're in a classroom, right? This is me. If I write something on the board, let's say, hi, and you write it down on your little notebook, you write down hi. You are transcribing what I wrote on the notebook. You wrote down hi in your notebook. But let's say, right, let's say that you have a friend who doesn't speak English. They speak French, for example. Can they read this? I mean, maybe, right? But maybe they know absolutely nothing about English, so you have to translate it for them, right? And you would do that by just simply taking what's in your language and translating it to theirs. Or I guess more accurately it would be Salut. Anyway, we go from hi to bonjour, right? So when you transcribe something, that is obviously the act of writing something down, where you're copying it down almost exactly. I mean, of course, you have your own handwriting, right? And then when you're translating it to someone, you're turning it into something entirely different, right? So when we transcribe something in biology, right, it's the same principle, right? So we would have to take like for example a DNA sequence. Let's say ATTCCG, right? Let's say that's a 3' end, that's a 5' end. And then to turn that into an mRNA, we would just do this. We would do the base pairs, right? So base pair rules, right? These are your base pairs in DNA. A, T, C, and G in DNA, and then A, U, C, and G in RNA, right? So always, A pairs with T in DNA, A pairs with U in RNA, and vice versa, and then C pairs with G in all the examples, right? So these are your base pair rules. Very important to remember, very important to know, right? So if we're taking this DNA strand, this is DNA right here, and we're copying that into an RNA, notice how we're still in the nucleotide language, right? But notice how we're changing from T's to U's, right? So we go A, T, T, C, C, G, and then we do the base pair rules. It would be U, A, A, G, G, C. Whereas, for example, if we were just reading off our DNA strand, let's say this is now our DNA strand, it would read TAA CCG. Right? But that's not the case because we're doing an RNA, and RNA is it's still nucleotide but slightly different, right? Just like how I have my own handwriting, and when you copy it down into your notebook, you have your own handwriting. So, for example, I would, I don't know, write in cursive, for example, and you would write in print. Well, print would be the prints would contain the U's, and my cursive would contain the T's, or whatever. Like this is the best analogy I could come up with, right? So furthermore, when we go into translation, right? So we just went over transcription. When we go into translation, we now take these nucleotide sequences. We now take these nucleotide sequences that we transcribed during transcription, and we're going to turn that into an amino acid, right? So we're changing language. So we're going from nucleic acids to amino acids, right? So that's the very basis of translation, right? So as you may remember from your from when we talked about organelles, we have what's known as the ribosome. So now the ribosome has two parts to it. A small part and a big part. Now these parts are very creatively known as the small subunit and the large subunit, right? So the ribosome, that's where we do all of our translation. The ribosome contains enzymes, and it contains like spots for an mRNA to bind, in which it could then facilitate the transfer. It could facilitate the construction of a polypeptide from an mRNA. It's really quite miraculous how this happens, right? So we have our mRNA. So this is our mRNA strand, and as you may know, mRNAs are single-stranded, right? So we have a ribosome, and then a ribosome kind of looks like this, right? It looks like a centipede, doesn't it? Our mRNA goes ahead and binds here at the ribosome, therefore kicking off translation, right? So translation has 3 steps. We have the initiation, we have the elongation, and we have the termination. So, yeah. Basically, how this goes, right, is normally the ribosome is actually dissociated into 2 parts, right? So this is prior to initiation, right? So this is our small part. I guess we could draw a little smaller, right? This is our small part, and this is our large part. And the large part, the large subunit of the of the ribosome has 3 constituent parts in it, right? We have the A site, the P site, and the E site. And we'll go over that in a second and what those all mean, right? So during initiation, our mRNA is going to bind to the ribosome until we reach a specific 3-letter sequence. Specifically, it has to be A, U, G, right? So always, your proteins are going to start with AUG. And AUG codes for methionine, right? So now we're going to introduce something called an anticodon, or an anticodon and a tRNA. So we have our mRNA, right, which stands for messenger RNA, and now we have our tRNA. Now we have our tRNA. And the T stands for transfer, right? So tRNAs look something like this. Let's see if I can draw it. And, okay. So this little 3-prong thing at the end, that is known as the anticodon. And up here, we have the amino acid. So the anticodon has 2 major parts, the tRNA has 2 major parts, the anticodon and the amino acid, right? So basically, we have the codon. So these three-letter sequences on an mRNA are called codons, and then we have the anticodon, which is also a three-letter sequence, right? So this, for example, this codon would read AUG, and since we care about polarity when we're talking about nucleotides, right? We have 5 prime, AUG 3 prime, and then our anticodon, since we're going to go off base pair rules once again, our anticodon will read 3 prime UAC 5 prime. And just like that, the base pair rules line up. So our anticodon is going to go ahead and bind here. We're going to go ahead and bind here at the codon. Let's see if I can do this. Just like that, right? And this act, the act of binding the anticodon here, will now make the large subunit of the ribosome bind to the small subunit, right? So yeah, let's move some of this. Let's move some of this over here. Yeah, that looks good. So now, this is all happening. This is all step 1 of the operation. Initiation. So now we're going to go into elongation, right? Where now we have the whole setup, right? And I'm just telling you this all for your information, right? I think it's important to know how this actually works in order for you to fully understand it, right? So we have these 3 sites that we cared about, right? The P site, the A site, and the E site, right? During initiation, when we bind the anticodon, we're actually going to start at the P site, right? So we're just going to draw it as like a little boot now, right? Right, so let's consider that to be our boot, right? So now we have the amino acid here, and so here's where I finally tell you what these letters mean. Okay, here's what they actually stand for. A is aminoacyl, P is peptidyl, and E is ejection. Here's what I think it stands for. A is for accepting, P is for processing, and E is for ejection, right? Because here's what we're going to do. We're going to take another tRNA, and this other tRNA has an amino acid on it. We're going to take another tRNA, and that's going to bind at the A site. See, just like that. It's going to bind at the A site, right? So now, since this binds at the A site, what's going to happen is this amino acid will be transferred over here, right? So we can go ahead and erase this. That doesn't quite work, does it? We can go ahead and erase this. So the amino acid will be transferred over here. So now we have a growing chain of amino acids, and then when the ribosome shifts over, all of our tRNAs therefore shift over 1, right? So now our ribosome is going to look something like something like that, right? That's a very crude drawing, I know. But look what happened. We had our tRNA here, we had another tRNA here, right? And then once it all shifts over, we have this tRNA at the A site moving into the P site, and then the tRNA at the P site moving into the E site. And since the E site just simply ejects the tRNA, it is no longer used, right? So imagine this happening over and over and over again until we have a very, very, very long chain of amino acids, right? Look at that. Look at that. And there's an alpha helice, there's a beta sheet, right? Yeah, just like that, right? So now, when we get into termination, right? So termination's a bit of a, whoops, termination's a bit of its own thing, right? So now, once we get into termination, we are going to have a stop codon. So stop codons don't actually have an amino acid. A stop codon will now bind at the A site, right? A stop codon will bind at the A site, and that'll just halt. It'll just halt all translation. It'll just make it stop. It counts as what's known as a release factor or a termination factor, right? Where so now we have our amino acid chain. Now our amino acid chain will go ahead and go away, right? Our amino acid chain goes away. It is released. Our translation is terminated, and not only that, but our ribosome is disassociated again, and our mRNA is also let loose, just like that, right? So that's termination. So a termination factor would be a stop codon, right? And there's a few different stop codons. I think UAA, UUA, UUU. I'm not 100% sure. Don't quote me on that. But yeah, that's termination. So initiation, elongation, termination. That's just the basics of how it all works and how you would visualize it. Well, it wasn't entirely necessary to do. I still hope that was a useful explanation how all of this works. Right. So what is used as a template for protein synthesis? It's not going to be tRNA. It's not going to be tRNA. It's not going to be microRNA. Your template for protein synthesis is going to be an mRNA. Right. So when we're talking about translation, we also have to talk about what a polyribosome is. Right. So a polyribosome essentially just means, well, I was thinking about this anecdote earlier, right? There is a cocktail known as the Ramos Gin Fizz. Now, the Ramos Gin Fizz is a notoriously difficult cocktail to make, or it's a cocktail that's difficult to make on a busy night because it takes a lot of time, right? So what would essentially happen is you'd make the cocktail, and then it would require a lot of shaking. So you would pass the shaker to one waiter, pass the shaker to another waiter until it's ready, right? In a similar principle, that's what a polyribosome is, right? So we have our mRNA. So this blue thing is our mRNA, and look at how we have many ribosomes, right? These many ribosomes can be considered our waiters, right? And the mRNA can be considered the drink, or the shaker, or whatever. I don't know. The analogy is falling apart. Anyway, so look what's happening here, right? On the same mRNA, we're making multiple copies of the same protein, right? All these ribosomes are just facilitators for this. This mRNA contains all of the same information. Nothing changes about the mRNA, and since nothing changes about the mRNA, the protein will be the same. See, look, we have this little like pink, blue, and white motif. Look, we have that here, 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 and here, right? We're just making it longer. We're just gonna make the exact same protein multiple times over, right? So notice how this one over here has been going on the longest, so therefore this one's the oldest. This one's been going on the shortest. This one's therefore that one's the youngest, right? So that's essentially what a polyribosome is. That's another thing you have to know about translation, right? And we also talked about what the first amino acid in new protein is always going to be. We should know that it's always going to be AUG, translating into methionine. AUG, remember this, AUG makes methionine. Met. We love the Mets. I don't know if you do. I don't really follow baseball. AUG to methionine, right? Methionine, this is your start codon. This one always starts first. You will always see this one first. Methionine is always first, right? So remember that. Anyway, we talked about an anticodon, right? What that does. We talked about translation. We talked about termination, what that means, right? So yeah, yeah, we went over just about everything. So I'm gonna introduce a bit of a practice, perhaps, right? So let's say we're carrying a tRNA, right? So we have a tRNA, and this tRNA's anticodon reads 3' CAC 5'. So what will the mRNA read? What will the mRNA codon read? Because this is our anticodon, right? What will the mRNA codon read? It will read GUG, 5' to 3', right? That's what will happen. So we can now determine what amino acid this codon will be carrying. And how can we do that? And I just realized I didn't even teach you how to translate. So how do we do that? We take a special little thing, a special little reference that you will get on your exam, known as none other than the codon chart, right? And this was a clip art, so you can't really see the text super well. Basically, you use this side first, this side second, and this side third, etc. Right? So let's see what we're working with here. GUG, right? So what is GUG? Let's use red. So let's see. Number 1, G, so that's what we're working with. Something along this line. And then U, okay, so now something along this line. And then we could find, alright, there's our last one. GUG, right? So we match all the intersection points. That's how we find our codon. So we will see now that GUG codes for valine. Right, and as you may notice, a lot of these codons code for the same thing. That's just it's redundant for a good reason, because like let's say you have a mutation from AAU to AAC. Well, if you have that mutation, then it doesn't actually change it. So that redundancy is very important for maintaining fidelity in protein making, right? So later on, I'll give you another example of exactly how to do this, how to translate, but that's just the basics, right? So if we're going from a tRNA to an mRNA, which you will do, we can go from a codon. So we have these 3-letter sequences. We can go from a codon to an amino acid, right? So moving on. So, as a matter of fact, why don't we just go ahead and do it now, right? Whoops. There we go. Okay. So let's take this this sequence here. And we're also going to use our little codon chart, our little cheat sheet, if you will, even though it's not really a cheat sheet. If anyone tells you you need to memorize a codon chart, that you're trying to deliberately waste your time, believe me, learning it is not worth your time. Just use the resource, right? So let's see what we have here. Let's see what we're working with here. So I've even gone ahead and split him up into 3s. Very kind of me, I know. Right? So let's go ahead and start. Right? So AUG, we already know that one. That's going to be methionine, right? GCU, what's that going to be? All right? So GCU. G over here, C over here, and then so GCU is going to be alanine. How about ACU? So, whoops. Once again, same principle. ACU. We start with A over here, C over here. So we intersect. ACU is going to be threonine. And by the way, you don't need to know their names. You don't need to know the amino acids names. It's if you're a biochemistry major, you will at some point, but don't worry about it right now. For now, it's met, ala, and thr. So GAG, what's GAG? G, A, G. That's going to be glutamine. Sorry, nope. It's going to be glutamate. GLU. UAU. UAU is going to be tyrosine. And UAA is going to be a stop codon, right? So a stop codon doesn't actually code for anything, so we just stop right there. So how this would look like would be met, alanine, threonine, glutamate, tyrosine, and then you just stop right there. Right, so this is our short polypeptide, right? Met, ala, thra, gla, and tyre. Don't write stop. It's just, you just stop there. You don't write anything. So now we can get into some mutations, right? So there are two types of mutations, right? We have point mutations and we have frame shift. Point mutations mean that a single nucleotide has been changed, right? So if we go from AAG, for example, that will change into, let's say, AGG. Right, so point mutation is when we change a single nucleotide in a sequence, right? Just one. And then a frame shift can be one of two things. It could be an addition or a deletion, right? So let's say we have AAG again. If we're adding a nucleotide, we can now say it's like we can now say it's AUAG, right? And then over here, we can go from AAG to simply AA, right? So as you might imagine, frame shift mutations, right? You're literally shifting the reading frame, right? So let's say we delete one of these codons, then every single amino acid after it's going to be different, right? So that's a frame shift. Or if we add, same principle if we add another one, it's going to change our three-letter sequences. It's going to change the reading frame, hence frame shift, right? So addition and then deletion, right? And then point mutation, we're just changing a single point. And then for point mutations, right? For point mutations, we have three different kinds. Let's move up here. We have three different kinds. We have silent, we have missense, and we have nonsense. So a silent mutation is when it's a mutation where if we're changing a nucleotide, it doesn't result in a change in the amino acid. So we're changing the nucleotide, but the amino acid doesn't change, right? So that goes back to the redundancy of the codon chart, right? So CCU to CCC, since those both code for proline, it doesn't change anything and is therefore a silent mutation. It's silent because there is no actual physical manifestation of that mutation, right? So missense is when we have a change in the nucleotide sequence. and then the amino acid does change, right? So an example of that would be if we had CAU, which codes for histidine, into CAA, which codes for glutamine, right? So when it does change, that's a missense mutation, right? Now, for nonsense, we have a change in the nucleotide, but instead of the amino acid changing, it now codes for a stop codon. Right? So that would be like if we went from, let's say, UGU, which codes for cysteine, to UGA, which codes for a stop codon. Right? So that would be a nonsense mutation. So let's give this example. Right? This is from my review worksheet, if you manage to look at that. So if we go from, for example, over here, if we go from ACU, if we change that into ACC, what kind of mutation is that? Well, let's see. Since ACU codes for threonine, what does ACU code for? Or ACC? It also codes for threonine, so that would be a silent mutation, therefore. Right? So how about if we change how about if we change GAG, GAG, to GAU, GAU? What kind of mutation would that be? Well, let's see. GAG, which codes for glutamate, GAU, which codes for aspartate, right? So that would be a missense mutation. So now, UAU, okay, well, what does UAU code for? Tyrosine. Now, what if we change that to UAA? Right, we already know this one. It codes for a stop codon. So if it changes to UAA, it is a nonsense mutation, since it now codes for a stop codon. It ends early. If it ends early, it is nonsense. If it changes the amino acid, it is missense. If nothing changes in the amino acid sequence, it is silent. So these are all your mutations and the examples of. So, like I said, important to remember. These things will come up on your exam. So yeah, study those up. One last thing about translation. So you may be asking, right? We have a tRNA, right? If we have a tRNA, how in the hell do we get those amino acids on there? Well, we get them on there through a special thing called an amino acyl tRNA synthetase. Amino acyl tRNA synthetase. An amino acyl tRNA synthetase adds an amino acid to a tRNA. Here's what you have to know about them. There are 20. How many amino acids are there? There are 20 amino acids. So for every amino acid, there is one amino acyl tRNA synthetase. So 20 amino acids, 20 of these synthetases, right? Pretty simple, right? There's not 64, there's 20, right? We don't do it for every codon, we do it for every amino acid. That'll, if you take that to heart, if you pay attention to what I just said, here's a little secret for you. That's a free point on the exam. So, yeah, there you go. Anyway, so we talked a little bit about mutations and genetic effects, right? So let's go move on to gene regulation, right? So I'm not too fond of gene regulation. It's super important. Don't get me wrong. Super duper important, right? Still not my favorite thing. So let's go ahead and go over it anyway, right? Because we have to. So of course, gene regulation, but we start in the DNA. So we start in the DNA, right? So DNA, here's a little secret, is negatively charged, right? So a good way to tightly pack up DNA is through something called a histone. So a histone, let's draw that in blue. I like to imagine them as blue. Histones are positively charged tetramers. I believe they're a tetramer. It might be an octamer, actually. Hold on, let me look that up. Okay, so I lied. It's actually an octamer. Okay, so okay, well, okay. Let me see what I can do here. Imagine these are behind. Anyway, histones are positively charged. They are octamers, meaning they have 8 subunits. Point is, they're positively charged. DNA is negatively charged. Do we see where we're going here? Right? So DNA, what's going to happen is DNA is going to go ahead and coil up to these histones, and it'll go on its day. Right? So if you stretch out one of your chromosomes, here's a little secret for you. It'll go to 2 meters. 2 meters of DNA. That's a lot. That's very long. So how do we condense that? Through histones. Right? So now, for gene expression, there's a couple things we could do. Right? Since DNA is negatively charged, we could do a little something called acetylation. Right? So an acetyl group looks like this. Right? Wow, would you look at that? That's a negative charge. Right? So, if we put this acetyl group and we stick that on our histone, right? Whoa, what is that? That was weird. Okay. Okay, so if we go ahead and stick that acetyl group onto a histone, well, then we're neutralizing some of that positive charge, right? And therefore loosening up the DNA, right? So here's the thing, right? DNA by default is very coiled up. And that's for a reason, right? Because by default, all of your genes are considered to be off, which is a really good thing. Because if all of your genes were on in every single one of your cells, well, you'd just about explode. That's all I could say. You'd just about explode. You really don't want all of your genes to be expressed at once, right? You really don't want stomach acid in your eyeballs, nor do you want liver cells in your brain, or yada yada, right? Cells have distinct purposes, and it is through gene expression that these purposes are manifest, right? So by default, these genes are usually considered to be off, but there has to be a certain way in which these genes can be accessed, right? And one way is through acetylation, right? So acetylation is going to loosen up the DNA because we are neutralizing some of this positive charge on the histone. It's going to loosen up that DNA. We can maybe loosen up the DNA versus earlier. Remember, we coiled it around a few times. Now with the acetyl group, we're just going to go around once, right? So if we're loosening up the DNA, we're making it more available to be transcribed, right? So DNA acetylation is one manner of such a thing, right? It is one manner of gene expression, right? So that's one. So when DNA is packed tightly, we're not really going to be transcribing. When it's loose, we're golden, right? One more thing. Acetylation is a reversible process. So we can go ahead and take this acetyl group and we just go ahead and remove it when we don't need it. Right? Just like that. Pretty simple, right? Like I said, reversible process. So that's on your histone, right? So histone proteins can also be moved, right? These can be moved. So imagine it's here, now it's over here, right? So depending on where the histone is, depending on where the packing is, we could go ahead and change the way or change the amount of expression that there is. Right, one more thing is methylation. So DNA methylation is another method, right? So DNA methylation is where we literally go on the DNA and we add a methyl group, right? And methyl groups kind of act like a flag, right? Let's say we have our ribosome. Ribosome is orange. When a ribosome moves along, it is going to bump into one of these methyl groups and is going to have a very hard time of transcribing. So methylation is a form of negative control, right? And methylation can happen randomly. Methylation and acetylation are part of your what's known as your epigenome. So your genome is just simply your DNA. Your epigenome is all the stuff on top of it, right? So methyl groups, acetyl groups, your histones, all of that stuff, right? And epigenetics is not my strong suit, but it is a up-and-coming field and is very, very important for determining genetics and how certain environmental factors can affect us, like smoking. Smoking will create a lot of this DNA methylation, right? So yeah, that's just one example, right? So yeah. But the thing about DNA methylation, right, is that you have to remember this: it does not allow access for DNA like histones do, or sorry, like acetyl groups do. It does the opposite. It denies access to DNA for transcription, right? So, like I said, just remember that, right? So yeah, that is more so for your epigenome, right? So let's go ahead and ignore our histones for now, right? So within our DNA, right? Within our DNA, we have what are known as enhancers, right? So let's make our enhancers blue. So let's say over here on this side of the DNA, this is our enhancer, right? So an enhancer is kind of exactly what it sounds like. An enhancer enhances transcription, right? It essentially acts like one of those guys you see on the street every now and then that have the big old signs that are pointing you to their business. It's exactly that, right? Let's take our ribosome again, right? Our ribosome is going to... That's not a ribosome. I've been saying ribosome this entire time, right? When we're talking about DNA, ignore ribosomes. We're not talking about ribosomes. This is called an RNA polymerase. RNA polymerase, as the name entails, is going to copy from DNA and make an RNA strand from it, specifically mRNA. So our RNA polymerase is going to see the enhancer, it's going to really want to bind to DNA, and then it's going to bind here, and then just go ahead and do its thing. So an enhancer will go ahead and increase transcription, which makes perfect sense, right? That's a first for biology. Usually these things make zero sense, right? So that's a nice change of pace, right? So yeah, when we're talking about enhancers, they are DNA sequences. Remember, DNA sequences, not proteins, that increase the likelihood of transcription. So there you go. So yeah, more on transcription and gene regulation and expression. Um, we can now move on. So that enhancer that we just talked about, that was in eukaryotic cells. We can now move on to prokaryotic cells. We can now move on to prokaryotic cells. And prokaryotic cells have regions within their DNA known as enhance. Sorry. See, I'm not too familiar with prokaryotes, right? So give me one moment. Right. So prokaryotic cells have what's known as a promoter region. So we could go ahead and draw the promoter region in green over here now. So at the promoter region is a sort of form of more regulation in this sense, right? So promoter region can, instead of detecting RNA polymerase, it can detect a different type of protein, right? And this protein is known as an activator, right? It's known as an activator. So activator, as the name might entail, will bind to the promoter region, and it will uptick transcription, right? And this is kind of only in RNA. Sorry, this is kind of only in prokaryotic cells. Again, I'm not too familiar. I'm just telling you. What you need to know. But anyway, this sort of promoter region will bind this activator protein, which will then increase transcription. And with that in mind, we can now just start fully talking about the operon. Not any specific operon so far. We're just going to simply talk about the operon. What is an operon? Whoops. This is going to look terrible. Yep. Okay, let me find another example. So this right here is a very general structure of an operon, right? So operons are exclusively in prokaryotic cells, right? Exclusively. So these operons will have distinct regions, like the promoter, which we just talked about, the operator, and then the structural genes, which will go ahead and make an mRNA. And it is through this mRNA that will make multiple proteins, right? So we have two types of operons that we care about. We have inducible operons, and we have repressible operons. Right? So I don't really want to go into too much detail because, quite frankly, you don't need to know too much about these bad boys. You just have to know a little bit about what specifically a repressible and an inducible operon is, right? And what a relief, right? Operons are such a pain in the ass, aren't they? But anyway, we're still going to talk about them. Let's start with an inducible operon. And your inducible operon is going to be the lac operon, right? The lac operon. What better than the lac operon, right? Let me see if I can find a good example for you. Because trust me when I say I do not want to write down the lac operon. Okay, this. Oh, whoops. This is good enough. Right, so let's look at this image, right? So we have our region here. We have a region here that's actually going to be making an mRNA and therefore proteins. We have our promoter sequence and we have our operator. So basically what's going to happen here is that we have what's here known as a repressor. I'm sorry, like I said, operon's really not my strong suit. Right, so this is our repressor protein, right? And we have lactose down here. And here's a little secret. It's actually not lactose. It's an isomer of lactose. But for simplicity, we're just going to call it lactose, right? So normally, ordinarily, the lac operon is turned off, right? Which is why it's inducible, right? So lac operon is inducible, right? Meaning that it's normally turned off. So why is that important, right? Lactose. What is lactose? Lactose is a sugar. You need to break down sugars, right? So the thing about lactose versus glucose, right, is that glucose is a far, far, far more abundant sugar source than lactose is, right? So why would you make the enzymes to break down lactose, which is what the lac operon makes? Why would you make those enzymes if lactose isn't present? Right? So hence, the lac operon is quote-unquote inducible, right? And that's what this image is trying to tell you, basically, right? Ordinarily, this inhibitor protein or repressor protein, however you want to call it, normally binds to the operator. So if it's binding to the operator, it is going to quite literally I guess we have it over here, is going to quite literally block RNA polymerase from transcribing any of these genes. It's going to literally block it, right? However, right, if we add lactose to the repressor protein, if we add it, it is no longer able to bind to the operator, meaning that RNA polymerase can go ahead and transcribe these genes, right? So basically, that's just some background of what exactly that means, right? But once again, here's a secret for you. The only thing you have to know for the lac operon is what I already told you, right? And that is, in high lactose and low or zero glucose, that is when the lac operon is going to be expressed, right? In low amounts of glucose and high amounts of lactose, this operon is going to be expressed. And another reason for that, why I mentioned glucose, is because of the promoter region that I was talking about, right? So let me see if we can find a good image with this promoter region that I was just telling you about. So here's one, and this one, oh, that's way too small, so it's gonna look like shit. Yep. Okay. Let me see if we can find a better one. Okay, this is perfect. Alright, so at the operator, we're going to bind the repressor, right? We know that. We learned that just now. And we also have what's known as a cap site. And at the cap site, we bind what is known as CAMP. So if you might remember, CAMP is one of the most important secondary messengers for sugar, for breaking down sugar, right? So therefore, right, CAMP really only appears when CAMP really only appears when we have high glucose, right? So when we have a high amount of glucose, CAMP is going to not bind, right? Or I just want to make sure I'm saying that right. Right. So yeah, in times of low glucose, we're not making CAMP because CAMP will make a cascade that tells the cell to make a bunch of enzymes and to break down as much sugar as possible. That's what CAMP is for. So when we bind CAP and not CAMP, CAP essentially acts like a substitute for it. Again, I'm not 100% sure on it. Basically, when we're binding CAP, we have a low amount of glucose, right? And that's remember, that's what we're talking about. That's an activator, right? The CAP site can be considered to be our activator, right? And therefore, CAP can be considered to be our sorry, no. The CAP site could be our promoter region, and the CAP can be the activator, which we just talked about, right? So, like I said, just remember this. Low glucose, high lactose, lac operon is expressed. Makes perfect sense, right? Because we have lactose, and we need energy somehow, and we're not getting it from glucose, so, therefore, lac operon is expressed then. So, now we also have to talk about the trip operon. So the trip operon, the trip operon is an example of a repressible operon. Let me see if we can do this. There we go. That's not so bad, is it? Okay. So, this is our trip operon, right? So, what are we looking at exactly? Right? So, a trip operon, like I said, is an example of a repressible operon. So, what does that mean? Repressible operon means that it's always on until it's turned off. It's always on until it's turned off, right? So here on the trip operon, we have the TRPR, which is which codes for the regulatory protein, right? So that codes for the repressor, right? And the repressor binds, and it binds to the operator, and blah 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 blah, right? So in this normal form, when we're not binding tryptophan, this is tryptophan, by the way, tryptophan. When it's unbound, when tryptophan is not bound, it's not going to bind to the operator, and therefore it's not going to repress anything, right? So the ultimate goal for the trip operon is to make more tryptophan, right? But what if you have enough tryptophan? Are you going to make more tryptophan, or are you going to halt production? Well, you're going to halt production, right? So after we do all of this, right? After we do all this, we're going to make, I guess we could do a pentagon. We're going to make tryptophan, right? So we've made tryptophan, yay, we're happy. So now tryptophan goes back to the trip repressor, and it binds, right? So once tryptophan binds to the repressor, now we're going to bind the operator and then turn the gene off, right? So repressor operon always on until it's turned off, right? Like I said, simple stuff. You don't need to know the details. You just need to know a very, very specific thing, which is that when tryptophan is present, it causes repression of the operon. Right? That's why it's repressible. When tryptophan is present, it causes repression of the operon. Right? So, yeah. Anyway, and in general, right, if we have a repressor, and this goes for the lac operon too, so remember this, a repressor is always going to be a protein. And this repressor protein is going to bind to the operator. That says operator, whatever. The repressor protein is going to bind to the operator region. The repressor, which is a protein, write this down, is going to bind to the operator. Excellent. So, we have one more thing to cover before we move on to genetics. We have one more thing to cover. And that is alternative splicing. What is alternative splicing exactly? So, we take an mRNA sequence, right? Nothing out of the ordinary here. Take an mRNA sequence, right? And let's keep in mind, alternative splicing only happens in eukaryotic cells. Only happens in eukaryotic cells, right? So we're going to take our mRNA, right? We're going to take our mRNA. We're going to put it through this thing called a spliceosome, right? This is our spliceosome. Our spliceosome does exactly that. It's a protein that splices. Who would have guessed, right? And what's basically going to happen is we're going to take our mRNA sequence, which is going to be this box, for example, right? And I'll let you know what these A's are and what this little bracket is. We're going to take our sequence, right? We're going to cut it up into little bits, right? We're going to cut it up into what are known as introns and exons. Introns and exons, right? Introns go away. We don't care about introns. Exons are expressed. Exons are expressed, right? So we can consider this to be an exon, this to be an exon, and this to be an intron, right? So now what's going to happen is that we take our spliceosome, it does its magic, right? And it's going to cut out this portion of the ribosome and splice together what's left, right? Just like that, right? So now we carry things that are only expressed, right? So introns are coded from mRNA or coded from the DNA template. Introns are still coded, but they could be cut out using a spliceosome, right? However, right, this is the same mRNA, by the way. This is the same mRNA. Now, what if we consider this to be an exon, and this to be an intron, and this to be an exon, right? Same principle, right? We take the middle, cut it out, take what's left, and put it together, right? Like I said, this only happens in eukaryotic cells. This is how we could derive multiple proteins from just one gene, right? That's alternative splicing for you. If we alternatively splice exons, we could encode more than one protein from just one gene. From one mRNA that was derived from a gene, using alternative splicing, we can make multiple, right? So I hope that's clear. So just remember this, right? Because it trips me up. It used to trip me up. Introns do not stay in. Exons do not exit, right? Introns go away, and exons are expressed. So remember that. Exons are expressed. Exons are the ones that we care about. Introns go away. And I said that we would talk about this poly A and this cap, right? So this is the 5' end of the mRNA. This is the 3' end. This little cap is what's known as the 5' guanylyl cap. Guanylyl cap. Yeah. And this is and here in the 3' end, that's what's known as the poly A tail. Right, so basically what these do is they help protect the mRNA from degrading, right? Because it's a long and arduous journey from outside the nucleus to the ribosome. The mRNA is quite delicate and needs some protection. So how does it protect itself? Through the cap and through the tail. The tail gets cleaved off. We don't care about these A's. These A's don't code for anything, right? The A's don't code for anything. We just cut them off, right? Makes sense. And then we have the cap, which helps protect this side of the mRNA, right? So that's just what that is, right? They stay on. They don't get removed during splicing. So yeah. Anyway, let's talk some genetics. So genetics, right? You have a mom and you have a dad. And when mom and dad love each other very much, they make a child, right? Mom has her own sets of chromosomes. Dad has his own sets of chromosomes. And when you put them together, you implant that into a child, right? Mom has a gamete. It's an egg. It's big, very big. 400 times the size of a sperm. Dad has the sperm, right? These are both haploid cells, meaning that they only have one set of chromosomes. So when they come together to make a new cell, to make a zygote, they come together and make a full gamete. Not gamete. They make a full zygote. We take two gametes, put them in a zygote, right? So here's the thing, right? In genetics, we have what's known as dominant and recessive. Dominant and recessive, right? So dominant alleles, which we'll talk about alleles in a second, dominant alleles are more often expressed. They overpower, which is to say they overpower the recessive alleles. Recessive alleles are not expressed often. Which is to say, those are kind of terrible descriptions of what each one of those mean, which we'll go into more later. That's just a general description, right? So let's say mom has 2 chromosomes, right? Well, mom has 46 chromosomes if she's human, right? Mom has 46 chromosomes, right? 23 pairs of 2, right? So within each pair, remember homologous chromosomes? Remember when we talked about those? In homologous chromosomes, at the same spot, we'll have the same gene, right? But these genes can contain different information, right? So for example, let's say this is the gene for eye color, right? Let's say this one contains information for brown eyes. This one contains information for blue eyes, right? They're both, mom has both of them, right? Mom has both genes, but mom has brown eyes, right? Why is that? Because brown eyes are dominant. Brown eyes are dominant to blue eyes. So therefore, they will be expressed. However, mom still has the gene for blue eyes. It's just not expressed. So we would consider that to be, let's say B. Let's give it a B, right? Let's consider that to be B, little b. This is what's known as mom's genotype. So let's look at what a genotype is. A genotype off of Garvan Institute of Medical Research is the unique sequence of DNA, right? Unique sequence of DNA, right? It can refer to the alleles, the variants of, therefore, particular gene, right? However you want to say it. These are your alleles. Alleles are variations of the gene, right? So here we have the gene, right? And at these genes we have different alleles. We have the allele for brown eyes and the allele for blue eyes, right? But like I said, mom has both genes, but she still has brown eyes. So therefore, she is going to express brown eyes. This is mom's phenotype. Phenotype versus genotype, you can think of phenotype as in like physical, as in physical attribute, pH, right? Brown versus blue, right? She's expressing brown eyes, right? That's what this gene is, right? So yeah, dominance versus recessiveness, right? So what essentially this means is that mom can have two different variations of what her gametes look like. She could have a gamete that contains the gene for blue eyes, or contains the gene for brown eyes, or she could have a gamete that contains the information for blue eyes, right? So let's consider dad now. Let's say dad has blue eyes. So therefore, dad only has one variation of what the eye color can be. Blue eyes, right? Because remember, blue eyes are recessive to brown eyes. So therefore, dad has to have two recessive alleles in order for his blue eyes to be expressed, right? So mom and dad make the child, right? And we could predict what physical attribute the child has through what's known as a Punnett square. So, if you go back to elementary math, right, how would you calculate 11 times 11 without having known it, right? How would you calculate that? Well, we could do it like this, right? We could take each of its constituent parts and break it down. So, 11 is 10 and 1, right? And 11 is, again, 10 and 1, right? So we can move those over here to a little square. So 10 times 10 is 100. 10 times 1 is 10. 10 times 1 is 10. 1 times 1 is 1, right? So we're just crossing them. We're crossing them, right? We're crossing all of the possibilities to see what we get, right? It's the same principle, which of course 11 times 11 is 121. If we add all these together, we get 121, which doesn't really apply to a Punnett square. If we apply the same principle to biology, we could take these gametes, right? So these are potential gametes, right? This happens on a 50-50 scale, right? You could either have this gamete or you could have this gamete, because you have two sets of chromosomes, right? And each set of chromosome was split apart during meiosis, which we went over, right? And same thing with dad. But dad has a 100% chance of having blue eyes, right? But like I said, so this could be 11, this could be 10, right? We just cross them. We literally just cross them. We cross the possibilities. So mom's genotype, which is big B, little b, times dad's genotype, which is little b, little b. So now, using this square, we could go ahead and predict what phenotype or what genotype the child will have. So, just multiply across, right? Same principle. Big B, big B, little b, little b, little b, little b, little b. Right? So basically, now we take the amount of squares we have, which is 4, right? And we take the desired phenotypes. So we have 1/2. So we have 2 squares, so 2 divided by 4 is 1/2, right? 50%. So 50% will have blue eyes. And then 50%, because we're considering dominance here, right? 50% will have brown eyes. And just like that, that is the basis of Mendelian genetics, right? So let's go over another example, right? So let's say dad also has brown eyes, but he's a carrier for blue eyes, right? Like I said, same principle, just cross him, right? So we could have big B, big B, big B, little b, big B, little b, little b, little b. Right? So 3 out of the 4 squares have the dominant allele, so that therefore 75% will have brown eyes, and 1 out of the 4, so that's 25%, will have blue eyes. Can you even see that? I hope you can. Yes, you can. Okay, good. Okay. Like I said, that's the basis of Mendelian genetics, baby. Just cross them and you're good. You're golden. Right? So there are some other examples of a little more complex expression of genes, right? So let's take it, for example, a flower, right? Let's say a flower is red. And let's take another example. A flower is white. They're the same species of flower, right? Something has to determine these colors of their petals, right? Let's say red is dominant, so let's do A. Let's say red is dominant to white. Right? So this has a phenotype of white petals, this has a phenotype of red petals. So what happens if we cross them, right? Whoops. So when we cross them, we now have a genotype of big A, little a. However, big A, little a actually yields pink flowers. Whoops. Big A, little a actually yields pink flowers, right? This is what's known as incomplete dominance. So incomplete dominance is when we have 2 sets of alleles, and the dominant allele isn't entirely dominant over the recessive allele, right? So we have the dominant allele, which is big A, which leads to red, and we have little a, which is white. Incomplete dominance is when we have both. When we have both traits will kind of mix together and make something new, right? So big A, little a, make something different. That's going to be incomplete dominance, right? So we could go ahead and kind of reword this, right, such that what if we're crossing 2 pink flowers, right? if we're crossing two pink flowers, right? Now, as you can see, I'm just getting way lazier with it, huh? If we're crossing two pink flowers, what does that look like? We now have a 25% chance for red, a 50% chance for pink, and a 25% chance for white. Versus versus like I said, complete dominance, which it would be 75% dominant trait, 25% recessive trait, right? So, one more thing before we move on. Look at these genotypes, right? Look at these genotypes. They all have a distinctive quality to them, right? Two big A's, two little A's, a big A and a little A. These have specific terms to them, right? When we have, so let's say, for example, big A, that's going to be homozygous dominant. Right? Because, okay, so let's go over all the terms first. When we have big A, it is going to be known as heterozygous. When we have little a, that is homozygous recessive. Right? So homo means same, hetero means different. So homozygous dominant means that we have this big A. So homo meaning same, dominant meaning that we have both capital letters. Heterozygous meaning we have different letters, homozygous recessive, etc. Right? So now we can go ahead and move on, right? So when we're talking about gene expression, right? When we're talking about these actual genetics, right? I mean, you figure you take a biology class, only in the very last 2 weeks or so, you finally start learning about genetics. Anyway. We have in nature what is known as a wild type, right? So what is a wild type? Take polar bears, for example. Polar bears are naturally white, right? Polar bears haven't been brown for millennia, right? That is because polar bears have a mutation that changes their genes to read little a, for example. A wild type is essentially just an animal that's just out in nature, right? And or an organism that's just out in nature. A wild type does not necessarily correlate to dominant versus recessive. Wild type is just the most common traits that you see in any population. So if you have like a wild type allele versus a mutant allele, wild type is the one that's going to be natural. That's going to be normal, so to say. So polar bears that are white, right? It's natural for them to be white. You would expect a polar bear to be white. Versus a grizzly bear, you would expect a grizzly bear to be brown, right? Both contain their respective wild type alleles, right? But ultimately they are both wild type in that sense, right? Anyway, let's move on. So, one more thing, any individual can only have two alleles. You have two chromosomes, a pair of chromosomes, you can only have two alleles, right? That makes sense, right? I guess we could talk about quantitative inheritance, right? Like, for example, height. Um, Jeez, I'm sorry. I just lost all steam there. Right. Okay. So we could talk about quantitative inheritance. Right. Sorry, I don't know what happened here. Okay. Anyway. Okay. So we could talk about quantitative inheritance. That essentially just means that something like height or hair color or hair color or what's another good one? Skin color, skin tone. Quantitative inheritance just essentially means that there are multiple genes that factor into a given trait, right? For example, your skin tone is a multiple has multiple different factors to it. So does your height, right? You're probably not going to be exactly in between the height of your mother and your father, nor is your skin tone going to be exactly between your mother and your father as well, right? They both have multiple genes that are going to determine those genes. So quantitative inheritance, just think of like they have multiple genes. It's not simple Mendelian genetics in that case. They have multiple genes that contribute to these traits, right? So one more thing before I go, because I'm just about finished here. One more thing before we go is sex-linked inheritance, right? So as you may know, those who are male-bodied have XY on their sex chromosomes. Those who are female-bodied have XX, right? And these can vary sometimes. Sometimes you'll miss in a Y, sometimes you have an extra Y, sometimes you have an extra X. Those are all genetic disorders. Anyway, so here's the thing, right? Women have two X chromosomes. Men only have one, right? So when we talk about sex-linked disorders, we're talking about disorders that are distinctly on these sets of chromosomes, right? So let's take color blindness for example. Color blindness is a sex-linked X or sex-linked sex-linked on the X chromosome that is recessive, that is also a genetic disorder, right? So we just basically take our chromosomes, right, and we put a little superscript on them, right? So let's consider B, so, and then little b. Once again, we're using B's, right? Big B, little b, right? Big B determines just normal vision, and that's our dominant trait, right? Little b determines color blindness, right? So if you have the little b gene, it's going to be colorblind, right? But of course, the gene for color blindness is going to be masked by the big B, right? So if yours are both little b, that would mean a colorblind woman. If they were both big B and were big B little b, then no need to worry. However, for men, look at this, right? For men, look at this, right? X, Y. Men only have one chance to get it right. Big B or little b. Right? There's only one chance to have instead of having the buffer up here, right, that women do, men only have the one chance, right? So either colorblind or not, right? So we could take it to a Punnett square. So let's, for example, cross this exact pair, right? So let's take X superscript big B, little b, big B, and then Y, right? So if this was a family that was trying to have a couple of kids, right? What are the chances of having a colorblind child, right? So we have two chances of having daughters, right? And as we can see here, neither of them will be colorblind. However, one of them will be a carrier, which is important to note, right? Carriers of these genetic disorders do not actually exhibit the symptoms. They do not have the disorder. They still have the functioning allele that is necessary for a normal phenotype, but do not have the disorder, so are therefore carriers, right? So, anyway, moving on. So these are our daughters. Moving on to the sons. Right? So look at that. We actually have a chance of having a colorblind son, despite the fact that we have a normal color vision father and a normal color vision mother. Right? Because like I said, in women, they have that extra buffer for these X-linked genes. Right? They have two X chromosomes versus men, which have a Y. Right? So women have that buffer. Right? So overall, there's a 25% chance of having a child that will have color blindness, but there's a 50% chance of having specifically a son that has color blindness. Right? And we have a 50% chance of having a daughter who is a carrier, and a 50% chance of having a daughter who is not a carrier, and a 50% chance of having a son who has normal vision. Right? So those are sex-linked disorders, right? Anyway, I'd say that's just about everything. Before we go, I want to talk about microRNAs, actually. I forgot to mention them. MicroRNAs, right? They're pretty neat. I'd say so, anyway. MicroRNAs are a rather recent discovery, right? MicroRNAs are the only type of RNA that are used to regulate gene expression. Specifically, they could inhibit translation. They could inhibit translation, right? Or they could help degrade mRNAs, right? So microRNAs are a very important factor for gene regulation post-transcriptionally, right? However, on a list of types of mRNAs, or sorry, on a list of RNAs, microRNAs are your ones that are going to be the ones who do gene regulation the most, right? Anyway, I'd say I just about went over everything. So here's the deal, right? Like I said, this is gonna be my last video for a while. The old PLF gig, the old peer tutor gig, it's just about over for me, right? So it's been a real pleasure to be working with you all. If you're not even in this class, I'm glad for you to have watched. And if you got ads during this video, you could blame YouTube because I don't see a single cent of that ad revenue. Um, yeah, in any case, hope you all have a good exam. I hope you all have a good summer break, a good endeavor in whatever thing you choose to whatever thing you choose to chase down, right? I just wish you all the best, honestly, really. I care a lot for all of you, and I really do wish you all the best. So take care, and I guess this is Ethan signing off. So yeah, bye.